Hi everyone, today we're going to be solving AQA GCSE Biology Higher Tier Paper 2H. In this particular video, we are solving June 2019. Figure 1 shows a food chain in a pond. We can see algae, which are then fed by Daphnia. Daphnia are eaten by Hydra, and then Hydra are eaten by Dragonfly nymph. Algae, in this case, is the producer. Daphnia is the primary consumer. Hydra is the secondary consumer. And Dragonfly is the tertiary consumer. Since there is no consumer for the dragonfly in this food chain, so dragonfly is the apex predator in this food chain. Which term describes the Daphnia in the food chain? So it will be the primary consumer. Draw a pyramid of biomass for the food chain. Label each trophic level. So when talking about biomass, biomass refers to the biological material derived from living or recently living organism. And then the definition for trophic level will be a group of organisms within an ecosystem which occupy the same level in the food chain. So since algae is the producer, it will have the highest biomass, and then the biomass of Daphnia will be smaller than the algae, followed by Hydra, which will be even smaller than the Daphnia, and then Dragonfly nymph will be the smallest. Now we need to label it: algae, Daphnia, Hydra, finally Dragonfly nymph. Give one reason why the total biomass of Daphnia in the pond is different from the total biomass of algae. The total biomass of Daphnia will not be equal to the biomass of algae, mainly because not all of the algae that is eaten by the Daphnia will be digestible parts. Some will be lost in feces. Some other alternative answers will be algae, alright, not all of the algae will be eaten. Some can be lost in urine, and some energy can be lost in respiration. Students investigated the size of the population of Daphnia in the pond. To do this investigation, this is the method used. Collect 1 dmq of pond water from near the edge of the pond. Pour the water through fine net. Count the number of Daphnia caught in the net. Repeat steps 1, 2, 3, 4 times. So the steps 1 to 3 is repeated 4 times to obtain an average. The number of Daphnia in 1 dmq water can be seen here. The question says calculate the mean number of Daphnia. Calculate the mean number of Daphnia in 1 meter cube of pond water. 1 meter cube is given equals to 1000 dm cube. So first of all, we will do the average or the mean per dm cube. To do that, we will add all of the values and then we will divide by 5, which gives us an average of 14. So 14 Daphnia per liter of 14 Daphnia per dm cube of water. Now in 1000 dm cube, there will be 14 times 1000, so 14,000 Daphnia. The pond was a rectangular shape measuring length 2.5, width 1.5 and a depth of 0.5. So we'll have to calculate the volume. In this case, volume will be length times width times depth, and the mean number of Daphnia per dm cube is, per meter cube is 14,000. First of all, we're going to find the volume of pond, which will be 2.5 times 1.5 times 0.5, giving the total volume 1.875 meter cube. Then we will find out the number of Daphnia. 26,250. Since our answer has to be given in standard form, so we will turn it into 2.6, 3, 1, 2, 3, and 4. So it will be 2.63 times 10 to the power of 4. Rainfall can cause fertilizer to be washed from farmland into pond. The student investigated the effect of fertilizer on the population of Daphnia in water from the pond. The student put 20 Daphnia in each of the 5 different concentrations of fertilizer. The students counted the total number of Daphnia in each concentration of fertilizer after 2 weeks. We can see the concentration of fertilizer in milligram per dm cube and the total number of Daphnia after 2 weeks. At 5 milligram per dm cube, we get the highest number of Daphnia, which is approximately 275, and the lowest number of Daphnia occurs at 20 milligram per dm cube, which is approximately 35. A concentration of 5 milligram per dm cube of fertilizer caused a large increase in the population of Daphnia. Explain why. The fertilizer is acting as a nutrient source for the algae. So more algae means more Daphnia. More algae means more Daphnia, since Daphnia is eating the algae. Now, the answer can be written in this way. The fertilizer cost increased growth of algae, so more food for Daphnia available. So the Daphnia increased in number. Figure 1 is repeated below again. The population of Hydra will decrease when 20 milligram per dm cube of fertilizer is added to the pond. Explain why. A high concentration of fertilizer, as seen, a high concentration of fertilizer decreases the growth of Daphnia. Since Hydra eats Daphnia in this food chain, so a decrease in Daphnia will decrease the number of Hydra. So the answer can be written in this way. Hydra have less food because there are fewer Daphnia. Genetic material is made of DNA, which structure in the nucleus of human cell contains DNA. Human cell consists of chromosomes, and within this chromosome we can find DNA. So the answer will be chromosome. Figure 3 shows part of the one strand of DNA molecule. We can see the nitrogenous bases adenine, guanine, thymine. Now, X is labeling a sugar, so we will call it sugar. The specific sugar is deoxyribose. It is a pentose sugar, deoxyribose, because this is DNA molecule. Now, this is phosphate, so Z must be base. So it is the nitrogenous base. Only base is needed as the answer. Y is the whole molecule, so it will be nucleotide. We get these particular points from here. So that's why the answers have limited options. A complete DNA molecule is made of two strands twisted around each other. So the two strands twisted around each other in this way. What scientific term describes this structure? So this particular structure is called a double helix. DNA codes for the production of protein. A protein molecule is a long chain of amino acids. How many amino acids could be coded by the piece of DNA shown in figure 3? 3 bases codes for one amino acid. If we look back into the structure, we see a total of 3 triplets. So the answer will be 3 amino acids. Scientists have now studied the whole genome, human genome. Give 2 benefits of understanding the human genome. Understanding human genome will allow us to diagnose genetic disorder. It will also help us for the treatment of inherited disorders. And it can also help us understand evolution or ancestry or our ethnic origins. We can also trace human migration pattern using human genome. Only 2 of the points are needed to score the mark. Phototropism is a growth response by part of a plant to light. Name one other tropism. Give the stimulus the plant respond to in the tropism you have named. The other kind of tropism is called geotropism or gravitropism. And in this kind of tropism, the plant responds to gravity. Plan an investigation to show the effect of light from one direction on the growth of plant seedlings. Include detail of any controls that are needed. You may use some of the equipment shown in figure 4 and any other laboratory apparatus. So these are the apparatus that we are given: lamp, several pots of seedlings, scissors, ruler, cardboard boxes with lids. This question contains a total of 6 marks. So to answer this particular question, we must have a method that must lead to valid outcome, and it has to be sequenced in logical order. So the answer can be written as follows. Use several pots of seedling that will be given the same amount of water and the same temperature and same type of soil. Have one pot of seedling in an area where there is light from all direction, and have the other pot of seedling in a box with leads and a hole in one side with a lamp shining light from one side. Measure the seedlings' height at the beginning of the experiment by straightening them out against a ruler and calculate an average for each pot. Then measure again after 3 days using the same method. Calculate the mean height increase for each group, and then also use a protractor to measure the angle of bending and compare with the direction of light entry. We explained how phototropism in plant shoot helps the plant to survive. So when talking about phototropism, it is the plant's response towards light. So phototropism helps the shoot bend towards the light so that the leaves receive more light and more photosynthesis can occur, and the plant produces more glucose, or we can say starch, carbohydrate, or organic material. So these are the 3 points. The human eye can focus on objects at different distances. Figure 5 shows a clear image of distant object is formed in a person's eye. Explain how the person's eye could adjust to form a clear image of a nearer object. If we look into the diagram, we can see the muscles and the ligaments that are helping the eye to focus. So first of all, we will talk about the ciliary muscle. In order to focus on a distant object, the ciliary muscle must relax. And then comes the suspensory ligament. When ciliary muscles are relaxed, the suspensory ligament are pulled tight, so they tighten. When they tighten, the lens becomes flat. The light is less refracted, and as a result, the light can focus on the fovea. Explain how the person's eye could adjust to form a clear image of a nearer object. In order to view a nearer object, the opposite actions must be taking place. To observe a object that is distant, the ciliary muscle had to relax. Now, in this case, the ciliary muscles had to contract. So the answer can be written in this way. The ciliary muscles contract, so they have a smaller diameter, and the suspensory ligament slackens. The lens therefore thickens and become more rounded. We can say the lens is more convergent, meaning it bends light rays inwards more. The image is thus focused on the retina. Explain why a long-sighted person has difficulty seeing near objects clearly. A long-sighted person has eyeball which is too short. The lens cannot be thickened enough, or the ciliary muscles could be too weak, or the lens is not sufficiently elastic. So the light focuses behind the retina. Long-sightedness can be corrected by wearing spectacles. Describe how spectacle lenses can correct long-sightedness. Spectacle lenses can be convex or converging lens, which can be used to diffract the light rays inward more. This focuses the light onto the retina. Table 2 gives the classification of 4 plant species. We can see species 1. Alright. If we look into this particular data, we will see, alright, Table 2 gives the classification of 4 plant species. We can see there are 4 species, species 1, 2, 3, and 4. Alright, the question asks, species 1 and 3 are most closely related. What information in the table 2 gives evidence for this? Okay, so in terms of similarity, let's look at 4 of the species, how far they match up in terms of their similarities. So all 4 species are in kingdom plant. They are spermatophyta, all of them. But we can see species 1 and species 3 have a common class, that is they are monocotyledon. So species 2 and species 4, they are eliminated because they are dicotyledonous. Species 1 are have the same order, species 1 and 3. Species 1 and 3 does not have the same family, and from there they are different. So we will say species 1 and 3 have the same kingdom, phylum, class, and order. Figure 6 shows the inheritance of flower color in 2 species of plant. We can see pea plants and snapdragon plants. Parental generation, red and white. F1 generation are red. So we can see red is dominant, whereas the white is recessive. Snapdragon, red and white, produces pink. That means they are codominant. In pea plants and snapdragon plants, flower color is controlled by one pair of alleles. In figure 6, the parental generation plants are homozygous for flower color. In heterozygous pea plants, the allele for red flower is dominant. In heterozygous snapdragon plants, the allele for flower color are both expressed. Use the following symbol for the alleles in your answer to question number 5.2 and 5.4. What is the genotype of red flower pea plants in F1 generation? So since the F1 generation was homozygous and the red color is dominant, so it will be capital R, capital R. What is the genotype of a white-colored snapdragon plant? So the genotype for the white color should be CW, CW in both. So they should be homozygous dominant. A gardener crossed 2 pink flower snapdragons. Draw a Punnett square diagram to show why only some of the next generation plants had pink flowers. Identify the phenotypes of all the offspring plants. So the pink flower was CW and CR both at the same time. Since the pink flower plant comes from homozygous red and homozygous white, so they are all heterozygous CR-CW. So both of the parents will be CR-CW, CR and CW. The resulting The resulting offsprings are going to be CR and CR, CR and CW, CR and CW, and CW and CW. CR and CR is going to be red in color. CR and CW is going to be pink in color, and CW and CW is going to be white in color. The question asks, what percentage of the offspring would you expect to have pink flowers? So looking into the diagram, we can see there are two instances where pink colors have occurred out of 4. So 2 over 4 times 100 is equals to 50%. So pink flower would be 50%. Commercially, hundreds of pink flower snapdragons plants can be produced from one pink flower plant. Figure 7 shows a tissue culture technique used for producing many plants from one plant. Tissue culture technique uses the process of mitosis, which is a form of asexual reproduction. So the pink flowered snapdragon plant, a leaf is removed and scrape of several small groups of cells onto agar jelly. So the agar jelly is then given with nutrients and hormones, and then those were kept in sterile condition at 20 degrees Celsius. They turn into new plants, and then from those new plants, many snapdragon plants, all with pink flowers, are produced. So this is an example of asexual reproduction. No fertilization has taken place. Give a reason for each of the following steps shown in figure 7. Several groups of cells are scraped off the leaf. The main reason for this is to produce many plants. So we have to write so that many plants can be produced. Nutrients are added to agar jelly. Nutrients are needed for making protein or amino acids, or we can say for providing energy for respiration. Protein is also essential for growth. Hormones are added to the agar jelly. Hormones will allow the cells to develop roots and shoots so that differentiation can occur. The plants are kept in sterile condition. This is to prevent the entry of growth of microorganisms which can cause decay. So prevent disease or decay. The plant cells are kept at 20 degrees Celsius. 20 degrees Celsius provides like a optimum temperature for growth, optimum temperature for enzyme function. Explain why the method shown in figure 7 produces only pink flowered plants. All the new plants are produced by asexual reproduction, which is a form of mitosis. So all of them are genetically identical, or we can say they are all clones, or they are all CRCW genotype, or they all have the same genes or DNA. Water conservation is important to human body. Which gland releases the hormone that controls water loss from the body? ADH hormone is released by the pituitary gland. ADH stands for antidiuretic hormone. So it prevents diuresis or prevents large volume of urine. The other ones that are shown here, adrenal produces adrenaline, which is a fight/flight hormone. Pancreas produces insulin, which controls blood glucose concentration. Thyroid releases thyroxine, controls heat production by liver. Which hormone helps the kidneys to control water loss from the body? The hormone that helps the kidney would be ADH. We already talked about adrenaline and thyroxine in the previous question. Luteinizing hormone, or LH, is needed for ovulation. So thyroxine, we can say metabolism, and adrenaline, and we need it for energy as well as time of danger. A man is walking across a desert. The man has used up his supply of drinking water. Explain how the gland you named in question 6.1 and the kidneys reduce water loss. In a desert, the temperature is high and his supply of drinking water has been used up. That means the answer can be written as follows. Higher concentration of blood because less water in blood. This causes more ADH to be released. ADH causes increased permeability of kidney tubules to water. So, more water is reabsorbed from the urine. Some people have kidney failure. Doctors may treat patients with kidney failure by either dialysis or a kidney transplant. Explain to biological reasons why most doctors think that a kidney transplant is better method of treatment than dialysis. So in a kidney dialysis, the person has periodic removal of waste from blood. In a kidney transplant, the transplanted kidney always provides waste removal. We cannot refer to cost or convenience. So for the reason one. So the reason one will be changes in concentration of urea in the body is minimized. So there is a less chance of damage to the body cells. For reason two, we can say no need for repeatedly puncturing the skin or dialysis. So there is no chance for infection or no need to take anti-clotting drugs. No chance of blood clots. So no need to take anti-clotting drugs. Or we can say blood not in contact with machine. So no chance of blood clots. Ragwort is a weed that grows on farmland. Ragwort is poisonous to horses. Plan an investigation to estimate the size of a population of ragwort growing in a rectangular field on a farm. To get a full mark on this particular question, our investigation must produce a valid outcome and it must be logically sequenced. So the answer can be written as follows. Use a 1 meter square quadrant. Place the quadrats randomly by using a random number generator app or random calculator generated coordinates. Place the quadrant at least 10 times and count the plant number, ragwort plant number, within the quadrant each time. Then calculate the mean number of plants per meter square. Then find the area of the field by multiplying length with breadth, and then population is equals to number of plant per meter square multiplied by the area of the field. Now, there are more points in this particular investigation than needed for 4 marks. However, we must complete a full answer. So, even if you forget 1 or 2 points from here, you'll still get the full marks once you complete the answer. The herbicide glyphosate will kill ragwort and other weeds. Scientists use bacteria for the genetic engineering of crop plants to make the crops resistant to glyphosate. Figure 8 shows the growth of a culture of the bacteria in a solution of nutrients at 25 degrees Celsius. We can see time in hours and the number of bacterial cells in millions per cm3. In the beginning, we can see there is a lag phase because the bacteria number is not enough for the number to multiply fast enough. Then we can see a log phase. In the log phase, the number of bacteria multiplying is enough in number that their multiplication is visible in the graph. Then we have a stationary phase. At this phase, we have number of birds is equals to death. The question asks, why did the rate of reproduction increase between 2 hours and 7 hours? So between 2 hours, which is starting from here, and 7 hours, this is the time. Why is the rate increasing? Because there is enough number of bacteria so that the rate can increase. So we will say more bacteria, so more reproduction per unit time. After 12 hours, the rate of reproduction decreased. Suggest 3 ways the scientists could maintain a high rate of reproduction in the bacterial culture. We can If we draw a line at 12 hours, we can see the number of bacteria reaches the stationary phase, where the number of bacteria produced is equals to the number of bacteria that dies. One of the main reason, they said, suggest 3 ways the scientists could maintain a high rate of reproduction. One of the main reason we have reached a plateau is because the bacteria ran out of nutrients. So we will say adding more sugar, adding more protein, adding more oxygen, increasing temperature, removing toxins, or by stirring the culture. These are the ways we can keep the high reproduction rate. The rate of reproduction of bacteria is fastest at 7 hours. How many times faster is the rate of reproduction at 7 hours than the rate at 12 hours? To find the rate at 7 hours and at 12 hours, we will have to find gradient. To find the rate, draw a tangent on the point at example at 12 hours on the curve, and then rate is equals to gradient, which is equals to del y divided by del x, or rise over run. So the rate at 12 hours is equals to 150 minus 60 over 20 minus 4, which gives us 3.43. And when we're going to find out the rate at 7 hours, we can take 5 hours and 10 hours. So at 10 hours, it is 80. At 5 hours, it is 25. 80 minus 25 over 10 minus 5. This gives us 11. So the rate at 12 hour is equals to 3.4 and the rate at 7 hour is equals to 11. If we do the scale factor, the rate at 7 hour over the rate at 12 hours, then the answer comes 11 over 3.4, which is 3.2. So the rate at 7 hour is 3.2 times faster. Scientists transferred a gene for the resistance to the herbicide glyphosate into the bacteria. The genetically modified GM bacteria can then transfer the glyphosate resistance gene to a cropland. Explain the advantages of making cropland resistant to glyphosate. Here, the thing that we need to notice is that glyphosate is the weed killer. So if our cropland is resistant to the weed killer, then we can spray the weed killer so that we can kill off all the other competing weeds and our plants will have the best chance of growing and producing crop. So we will say this causes the glyphosate to kill the weeds but not the crop. So less competition for light, water, nutrients with the weed. And as a result, the crop plant will have a higher yield. It is important to keep the blood glucose concentration within narrow limits. A person eats a meal containing a lot of carbohydrate. This causes an increase in the person's blood glucose concentration. Explain how the hormones insulin and glucagon control the person's blood glucose concentration after the meal. Insulin is released in high glucose concentration in blood. On the other hand, glucagon is released when the blood glucose concentration goes low. So the answer can be written in this way. Blood glucose concentration increases after the meal. This causes insulin secretion and then insulin causes blood glucose to enter the cells, or we can say liver cells or muscle cells. The insulin then causes blood glucose that has entered the cells of the liver or muscles to be converted into glycogen. So blood glucose concentration decreases. This causes glucagon to be secreted later on. Glucagon then causes glycogen in the liver cell to be broken down into glucose and released into the blood, thus maintaining the person's blood glucose concentration. The body cells of a person with type 2 diabetes does not respond to insulin. A person with type 2 diabetes often has a higher blood insulin concentration than a non-diabetic person. When talking about type 2 diabetes, we have to understand this is a lifestyle-related problem. The persons with type 2 diabetes, the body becomes irresponsive to insulin. So now, in this answer, we'll have to explain why the person with type 2 diabetes often has a high blood insulin concentration than a non-diabetic person. Since the body is irresponsive to insulin, so more insulin is needed to achieve the same job. The answer can be written in this way. The liver or the muscle cells absorb less glucose in a type 2 diabetic person. So, the glucose concentration in the blood remains high, and the high blood glucose concentration stimulates the pancreas to release more insulin. Metformin is a drug used for treating people who have type 2 diabetes. Scientists investigated the effects of metformin and two other drugs, A and B. The scientists wanted to see how the drugs affected the blood glucose concentration of 220 people with type 2 diabetes. This is the method that is used. Put the 220 people into 5 groups. Treat each group with a different drug or a combination of drug for several weeks. Give each person a meal which is high in carbohydrate. Measure the blood glucose concentration of each person 30 minutes after the meal and again 3 hours after the meal. Suggest 3 variables that the scientist should have controlled in the investigation. Since the scientist is using 220 people in 5 groups, one of the first things the scientist should keep in control is their age. Age is a factor that can affect. Other than that, also the scientist should have height and mass of all of the groups similar, since body mass index affects the severity of the diabetes as well. Then, proportion of males to females. in each group. Other points that we can write, because this is a suggestion question, so there are other answers that we can write as well, other than these 3 points, which are severity of their diabetes, dose of drug, starting food, blood glucose concentration, or if they have any other health condition. So any 3 points out of the 7 points will be the correct answer. The scientists recorded their results as a mean value for each group. The scientists calculate the standard deviation for each group result. Standard deviation is a measure of the spread of individual results above or below the mean value. So basically, standard deviation is a measurement. If there is a mean value, how much higher it can lie or lower it can lie from the mean value. So let's say this is the mean value. So how much a data can lie above or below the mean value. If the standard deviation of a data is big, then that data is less reliable. If the standard deviation of a data is smaller, then that data is more reliable. The scientists gave each group result mean plus standard deviation. The larger the standard deviation, the greater is the spread of the result around the mean. Which of these result is the most precise? So the most precise will have the smallest spread below or above. So this one 177.2 plus 15.4 as the smallest, it's more precise. So understand the term precise here. Precision means how close together the values are. The smallest SD will be the closest value. Table 3 and figure 9 shows the scientists' result. The number of people metformin is 60 with 177.2 with plus minus 15.4 mean blood glucose concentration 30 minutes after the meal in milligram per 100 cl. And then with A, 40 people, we see 182 plus minus 18. With B, 25, 171 plus minus 16. Metformin plus A, 65 people in the group, 205 plus minus 19.4. Mean blood glucose concentration after 30 minutes of the meal. With metformin plus B, we can see a higher value. The lowest value from all of this data is achieved by B. However, the standard deviation is a little bit higher, and metformin, where the standard deviation is a little bit smaller. The mean percentage reduction in blood glucose concentration 3 hours after the meal is represented here. Now guys, we need to understand one small information in terms of if the data between one group and the other group is significant or not, how to know about it. If the data, which is represented here, has a standard deviation in any point, if they overlap, as you can see, all of these data are overlapping, okay, then we can say that metformin A and B has no significant difference because there's SD's overlap. In table 3 and figure 9, some standard deviation of results overlap. An overlap of standard deviation shows the difference between the means are not significant. No overlap of standard deviation shows a significant difference between the means. A student looked at the scientist method and the results in table 3 and figure 9. The student stated metformin works better than when used with other drugs. Evaluate the student's statement. So guys, the student statement, let's understand it. Metformin works better when used with other drugs. So let's see. So interpreting this particular graph, we can see with metformin, we have 32 mean percentage reduction in blood glucose concentration is 32%, with the standard deviation going up to 38 and going as low as 24. With the drug A, we have reduction of 33%, the standard deviation going as high as 41 and as low as 25. When using drug B, we get 31% reduction, but as high as 35 or as low as 27. But metformin plus A gives the highest blood glucose concentration reduction, which is 51%, or as high as 55, or as low as 47. Metformin plus B gives us 42% reduction, as high as 47 and as low as 37. So we can see with metformin plus A, we have a highest amount of reduction and there is no overlap almost. So we can say, by supporting the answer, we can say metformin plus A gives significantly greater reduction in blood glucose compared with MET alone, and this supports the statement. Then we can say for the metformin plus B gives a greater average reduction in blood glucose than MET alone. This also supports the statement. Now guys, one thing that you need to notice in both of these answering points is that in the first case, it is written metformin plus A gives a significant greater reduction. And in the second statement, it is written metformin plus B gives a greater average reduction in blood glucose. Why the second one is not written significant? So this answer lies on the graph. If we look back into the graph, we will see the problem with metformin overlap of the SDs occur. However, in terms of their average, we can see the average for metformin plus B is much better than only metformin. Hope you understood the difference. Now moving on for the third point, we will say metformin plus A standard deviation does not overlap with the standard deviation of metformin alone. So there is a significant difference. However, MET plus B SDs overlap with metformin SD, so difference is not significant. Now, we cannot conclude the data completely because the group sizes are small and not the same. So the results may not be representative of the original result. So we can give a conclusion at the end. The conclusion will be, conclusively, MET works better on average when used with other drugs based on these results. However, the data may not be reliable enough to validate this. Further investigations are needed. Guys, thank you for watching this particular video. Guys, subscribe to the channel so that the channel can grow, and question paper solutions like this will be uploaded nearly every day. So keep an eye for your favorite question paper, or write in the comments which question paper that you want. And eventually we will be uploading all question papers related to chemistry, biology and physics, all the British curriculum boards.